Hey everyone, I am going to go through um, the second part of our central dogma step, which is going to be called transcription, uh, the beginning portion where we're going to go and convert DNA into RNA so that we have that code later on to create some proteins. So I'm just going to start by recapping the differences between RNA and DNA so we have that visual. We know that all genetic material is made up of nucleotides, and those nucleotides are going to have a sugar and phosphate backbone, which we can see um, happening here. I guess that's a better visual. Where's my laser pointer? There we go. So here's our sugar phosphate backbone. I know that's not shown there, but remember our sugar is going to have a 5-carbon sugar ring to it. Our phosphate is one of our groups, which is PO4, and those are going to alternate to create that backbone. And then these colored sticks here are representing our bases, our nitrogenous bases, C's, G's, A's, and U's if we're talking about RNA, or C's, G's, A's, and T's if we're talking about DNA. So again, remember the double helix for DNA. RNA is a single strand. The sugar is different. RNA is ribonucleic acid, so its sugar is ribose, and then DNA is deoxyribonucleic acid, so its sugar is deoxyribose. Very similar, but just a slight difference to them. And then we can see that the differences between the bases are going to be uracil and thiamine. Again, very similar in structure and shape, which makes sense why they could kind of replace each other. But just there are slight differences in chemical composition. So there's the breakdown. We have talked about this quite a bit already, so I'm going to kind of just skip through this, but we're going to focus on this last piece down here, how RNA is going to be a temporary molecule for our genetic code. It's going to be super helpful in protein assembly because we can't take that DNA genetic material and bring it on out over to a ribosome. It's only RNA that does that because it can move um, more freely due to its single strandedness and this particular molecules like uracil that are attached to them. So um, here is our overview. Transcription is going to occur in the nucleus and it's going to utilize the enzyme RNA polymerase. So we know we're making RNA in this case, and it's going to make a long chain, which is the polymer part, and it's an enzyme, so ACE. It's going to take on its two jobs. It's going to take that um, highlighted um, active gene that needs to go through transcription and translation, and it's going to take that sequence of DNA and it's going to attach and unwind like helicase would normally do, but we only have our one enzyme this time. So RNA polymerase does the unwinding of the double helicase, of the double helix, and then also is going to build the chain of growing RNA nucleotides to it. So, how does it do that? It's going to have some major pieces. So, for all of transcription, there's going to be a region on our DNA called the promoter region, and it has these repeating T's and A's. It's called the Tata box, which is kind of funny to say, but it's going to be repeating TA, 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 and that's going to signal, hey, it's transcription time. So that's our recognition sequence for the beginning of that gene to be transcribed. And then it's going to go through and have that elongation process. And then there's also going to be a termination sequence at the end, um, kind of like a palindrome almost, but AAU, AAA. And again, once that's read, it's going to signal the end of um, transcription. Oops, I'm trying to get out of here. Sorry. I linked this into our notes region, but I want to play it for you too. So, So again, this looks kind of weird, but this molecule here that's in our spiral is our DNA, our genetic material in its helix. And then we've got some uh, larger globular molecules that are going to kind of initiate, right? Read that TATA sequence. And then we're going to have a growing mRNA transcript that's going to build off of there. So right now it kind of just looks like a ball of yarn, but so there are transcription factors, little molecules that assist in the processing. So that's what's happening where all those things were flying in. So that's our RNA polymerase molecule right there. And then we can see the messenger RNA. All right, so we're done with that. I'm going to go back here and continue. So we kind of saw that initiation promoter region where all those transcription factors were flying in and attaching before anything happened. And then once it was ready to roll, that RNA polymerase molecule was the last thing to attach and cause the whole processing. And then it'll eventually reach that terminator sequence, which shut it down. So again, the promoter is going to have some recognition sites, like we talked about that TATA box, so that it knows where to start. And then there's going to be a specific region of that genetic material that's going to be transcribed, not the entire set of DNA, because that would be super inefficient. So that RNA polymerase molecule, which we kind of see faded over here, is going to attach, create a replication bubble, and just open up a portion of the DNA while it's unwinding and adding in the bases. And then it'll reseal that DNA as it moves along. So again, here's our recognition sites, the TATA boxes. As those molecules are being attached in red, they will locate themselves in this region. We don't have to worry about the upstream marks of the 35 or 10 element. Just ignore that. So again, there's 3 steps always to every scientific process. The first one is going to be initiation, like we talked about, locating the initiation sequence, bringing in those transcription factors, and then opening up and unwinding the specific gene, activated gene. The next step is going to be elongation. So that RNA polymerase is going to pull in the complementary bases to that DNA strand. And it's going to make a single stranded chain of RNA. And it's actually going to be messenger RNA. So it's going to have A's, U's, C's, and G's in it. And why do we have U's in there? It's going to be a less expensive base to create. It's also got a very short term lifespan, which helps eliminate some of the issues where it could cause damage, or um, the DNA could be impacted by other molecules that could come and attach and cause some issues to it. So the U part just gives it a short lifespan for that strand of messenger RNA. And so our DNA has the T, which has a lot more stability to it, which we need because our genetic material is based off of that, right? So this is my, again, RNA polymerase that we saw in that video. Here's my growing messenger RNA strand. It's going to end with the termination sequence when it reaches the end of that activated gene. It's going to read a signal of AU, AAU, AAA, which are stop, stop. And then that pre-messenger RNA strand is going to separate away from RNA polymerase, and then it needs to be modified before it can leave the nucleus. So there's two parts. It's going to create a little cap or hat on one end where it's called a 5' cap or a guanine cap, where there's 3 to 5 G's that are added to the front to protect it and also identify this is the front of the messenger RNA transcript. And it's also going to make a long string of adenine nucleotides at the end called a poly A tail. And this poly A tail again signals this is the back side of our messenger RNA molecule, but it also determines the overall lifespan of that messenger RNA molecule. So the longer the tail, the longer the lifespan. There's some additional terms that I'm going to talk about next, which have some easy definitions to them. Introns are actually going to be taken out. They're the non-coding genetic material. Exons are the coding genetic material of our messenger RNA. RNA splicing is that process of kicking out the introns and gluing together the exons, and that occurs at the molecule known as a spliceosome. So here's a little visual of it. You can see our G cap and our poly A tails have been added, and now we have some non-coding genetic material here that needs to be removed from our RNA transcript. So that's going to be cut out. And then these remaining pieces are called exons. And that's our coding genetic material, that's going to be glued together so that we have just the information that we need and no extra junk involved. And this is the mature messenger RNA molecule, which is ready to leave the nucleus, has been processed and timed. So again, it's called a spliceosome, the location or the region where this happens, but basically it's just going to fold and separate. And the reason we care about this process is we could have all sorts of unique combinations of exons and removal of introns. Sometimes they leave a few portions of these introns in there, so there are unique situations for what's left over. And then again, they can fold in different patterns. So it's not just neighboring exons that are next to each other. They could pull in one that's later down the stream and move it up higher and kind of rotate them around at the spliceosome. So you have a really unique transcript of messenger RNA that is kind of cut and spliced together. So I have some transcription practice for you. Use our original DNA sequence, pair it with its complementary base of messenger RNA molecules. So A's go to U's, T's go to A's, G's go to C's, and C's go to G's. There's another one for you to do some reverse practice where you have the messenger RNA, and you can go to DNA. And then we talked about this already, how there's different versions of messenger RNA. So I just wanted to cover those pieces with you today. And we'll talk about these other types tomorrow as we go through translation. So there we have it.